Welcome to Unit 6, Lecture 2. This is a lecture about macro and micro evolution. So, Charles Darwin came and kind of helped put all of the pieces together to make us, or to help us understand that evolution occurs. Now, since Darwin, since Mendel, as we've moved on, we can now actually see evolution happening on a micro scale, which is a gene pool generation by generation, but then also a macro scale when we see big events happening. And so that's some of the stuff that we're going to cover here. Okay, so microevolution is actually just the evolution of allele frequencies. This does not always mean a new species. Okay, this does not always mean a change in species or we create a new species, but microevolution basically means that our gene pool is going to change. And changes in the gene pool can result in changes in a population and then changes in a species, but it doesn't always necessarily mean that it creates that kind of evolution. Two guys, Hardy-Weinberg, created mathematical ways to test this. So the Hardy-Weinberg equilibrium is a mathematical formula that helps us see when species are in equilibrium. There's two equations, p plus q equals 1, and p squared plus 2pq plus q squared equal 1. They basically mean the same thing. But it depends on what kind of information you have and what we're working with. So we have P, where P is the dominant allele, Q is the recessive allele, P squared is the homozygous dominant individual, 2pq is heterozygous, and Q squared is the homozygous recessive individual. So we can actually use information within a gene pool to actually study changes over the course of time. So here's a practice problem. So you guys are going to need a calculator. All right, we're going to work through this one together. Okay. We are going to do some more of these in class, but I want to work through one. So scale coloration in lizards has a law of dominance inheritance pattern where green is dominant to blue. So there are 1,024 individuals who are homozygous dominant, 512 individuals who are heterozygous, and 64 individuals who are homozygous recessive. So we need to find the frequency. So we're basically looking for all of these things. Okay, so the first thing we need to do, there, 2, 4, okay, 5, 12, and 64. Okay, so we need to total those all up. So 1,024 plus 512 plus 64 equals 1,600 individuals. Okay, so now, since we know it's 1,600 individuals, oh, that was fast. Okay, since we know it's 1,600 individuals, we can easily find p squared because 1,024 over 1,600 is going to give us our p squared individuals. Okay, so that should equal 0.64. Okay, our 2pq, and in this case, we can just do it. 512 over 1,600 equals 0.32. Okay, and q squared equals 64 over 1,600, which equals 64 divided by 1,600 equals 0.04. And if you add those all up, those should also be close to equaling 1. Hey, look at that. See, they equal 1 because p squared plus 2pq plus q squared equals 1. Okay, so that's the frequency, all right? And then to find the frequency of the alleles, 0.64, and if that equals p squared, if we take the square root of that, that should equal p, okay? So 0.64 equals 0.8. Okay, 0.8, and then we can just rearrange and do q. So if p plus q equals 1, 1 minus p should equal q, right? So 1 minus p should be 0.2. Okay, we are going to go over this more in class, but the biggest thing is to find the homozygous recessive individual or the homozygous recessive genotype, and then you can kind of work your way from there depending. Now in this example you were given all the possibilities, so it's not hard, but there are answers. Okay, so hey look, all my math was correct. Yay! To be honest, I don't have my answers in front of me, so I just worked that out with you. Okay. All right. So Hardy-Weinberg is a way for us to actually empirically measure whether a gene pool is changing or not. So there are some conditions that can affect Hardy-Weinberg equilibrium. There has to be a large population. The larger the better. We talked about this a little bit with rules of probability. If I roll a die 6 times, I should get every single number on that die. But with 6 rolls, it's probably not likely. If I do 600 rolls, it's probably a lot more likely that I'm going to get 100 1s, 100 2s, 100 3s, 100 4s, and so on. So the same thing happens with Hardy-Weinberg equilibrium. The larger the population there is, the more likely we will stay within equilibrium. There needs to be random mating, which basically means there's no sexual selection. There's no preference. Now, that would basically say that you walked out the door and the first individual that you met, you were like, all right, fine. Okay, you and I, we're going to have a kid. All right. In many populations, that doesn't happen. Okay, there's this thing called sexual selection where we talked about the peacock tail. The peacock tail is designed to make peacocks more attractive to peahens. Okay, that's its main goal, is to say, hello, I'm here. All right. And so random mating means that there's no sexual selection. Like I said, in most species that isn't true. There also should be no natural selection, no genetic drift, which means there's no movement of alleles, either through emigration, immigration, no bottleneck effects, okay, and then no mutation. So how likely is it for a population to be in Hardy-Weinberg equilibrium? Not very, okay? But knowing that it's not in equilibrium and knowing how to calculate equilibrium allows us to, like I said, show empirically that things are changing. Okay, and so that's why knowing that it's not in equilibrium is kind of just important as knowing when it's in equilibrium. Then there's macroevolution. So macroevolution are basically changes that are on a huge scale. So microevolution, we're talking just about a gene pool. Macroevolution, we're talking about changes to the entire, sometimes in certain cases, the entire Earth. Okay, so continental drift, plate tectonics, you guys have talked about that. That's where the plates just move. Earthquakes, volcanoes, all of that stuff is related to that. Mass extinctions. There have been 5 main mass extinctions. Some scientists would hazard a guess to say we are in a 6th mass extinction as we speak. We'll see. Only time will tell. Unfortunately, extinctions are, we see them after the fact, but that's mostly because we've only been, if we were to think about the Earth's lifespan as a calendar year, we have basically only been around for New Year's Eve. Okay, so there's a lot that's happened on Earth before we even showed up. So we're kind of late to the game. Okay, and then there's been cases where we see changes in developmental genes. And you can see in this picture right here, there's a group of genes called Hox genes, or homeobox genes, or homeotic genes. They basically determine our gene organization. And they're pretty conserved across animal species, all the way down from Drosophila, so the fruit flies that you guys have looked at this year, all the way up through us. These genes maintain a certain commonality from flies on up. But there are changes to their order Okay, there are changes in their gene expression, there are changes in how they turn on or turn off during development to end up creating multiple species. Then there's petamorphosis, which is where an adult body is maintaining what are known as previously juvenile features or staying juvenile for longer. So we've seen this in our own selves in relationship to a chimp. We look a lot more like baby chimps than adult chimps, and so we stay younger for a longer period of time, and that has to do with the fact that that allows our brains to actually develop more. When baby chimps are born, they have a little bit of time with mom, growing and then developing, but for the most part, they're off and running. Chimps, I believe, have an average intelligence of a 3 or 4-year-old. But what happens with being us able to maintain juvenile status for longer, we learn more, we grow more, our brains actually develop more. And then there have been cases where there's what's called a selective sweep. Lactose tolerance is a huge example of this. It's when a gene is so beneficial, or a gene change is so beneficial, that it basically just sweeps through a population without too much trouble. And so that's what happened, is lactose tolerance was so beneficial to northern latitude populations who couldn't farm all year long, that it became such a huge piece to the genetic puzzle that it just basically it swept through the human population. Okay. So most of the time, evolutionary changes are gradual. They're very small, they're gradual, we have little bits and pieces, but every once in a while there are events that create new species and huge changes over time, and generally what happens is that results in what's called punctuated equilibrium. So in this, you see, we start with an orange bird. Okay, something happened here that made yellow more advantageous, so that yellow bird survived and was able to kind of shoot off and become its own species. Something happened over here that created a red species, and then the orange species still existed. And so it's not where this happens where orange slowly changes, okay, but what we definitely see is we see huge events at one given time. Just keep in mind that when it comes to evolution, there is not a right answer or a goal. Okay, there is no end point in mind when it comes to evolution. Evolution is basically what's right for right now. Okay, what traits are good for right now, and then depending on how long those conditions persist, it'll depend on how long that population persists or can change. Okay. So wrap up. What is the difference between micro and macro evolution? Which is more likely to be closer related? Two species that look similar but with more variation in the genome, or two species that look different but with more similarity in the genome? Okay, so explain. And then rabbit's ears can be either short or floppy, where short ears are dominant to floppy. So there are 653 individuals in a total population, 104 floppy-eared and 549 short-eared. Find the frequency of the dominant and recessive alleles and find the frequency of the homozygous dominant, heterozygous, and homozygous recessive individuals. All right, I will see you in class. Have a great day.